And what you really want to do here, especially for connective tissue and maybe even epithelial tissue, you know, the whole idea is to sketch it, to make you learn, to pay attention to details. The problem with connective tissue is it's so obscure, it's really hard to draw. So what I would suggest is as you go over these images, you take screenshots and you make your own PowerPoint of it and then do this kind of label the structure stuff. So for any given sample, here are the slides in this histologyguide.com slide box. These are the very ones that I'm going to be using for the lab practical, the very ones, and I'm going to be asking you 4 things about them. What's the tissue? In this case, what's the cell, the fibroblast, or what's the fiber, right? And in this one, it's just what's the tissue or what's the cell, adipocyte. And this one, what's the tissue and what's the fiber and what's the cell, right? Or what's the tissue, what's the fiber, what's the cell. And you only have 4 right here. So that I want you to identify, but these will come up over and over again. So we want to make sure that you're able to recognize. If you do them, then you'll do well on the practical. If you don't do them, you won't do well on the practical. Otherwise, it's self-guided, but I'll definitely, you know, whatever if you're ever having a sticking point, then just bring it into class. The other thing I mentioned while I'm here, you could always present them, like show, hey, am I doing this right? Am I labeling everything that's relevant? Right? And I'll say, yeah, you're doing right, or yeah, you should do this or that, right? Take a different picture or something like that. The other thing, I got to re-edit this. These are from a different textbook. I'll put the appropriate figures so that you can kind of get a look of what the cartoon version is like in your Tortuno's text. I'll put those up after class. So this first one, we're trying to identify areolar or loose connective tissue. And so we're going to go to this histology guide. That link is up there. You go to the slide box right over here, and you're going to go to, in this case, chapter 3, connective tissue, right? And you're going to click on it, and there's going to be a whole bunch of different things, right? Have a little bit of a description, stuff like that. But you're going to want to find that MH260 sample. So we could look at one sample of loose connective tissue. So you'll scroll around. Oh, here it is, MH260. And notice that any sample, tissue sample, which is what we're trying to identify here, is going to be within an organ or a structure or something. I don't care that it's the mesentery, right? I'm not asking you to identify it as mesentery or a aorta or anything like that. We're just concerned about the tissue, right? So when I ask you what the tissue is, the wrong answer is mesentery. The tissue is loose connective tissue. And this picture looks just like your cartoon pictures that you have in your book almost. Right? You've got all these purple dots right over here. For our purposes, right? I'm going to show you exactly this picture on the practice. Okay, I'm taking screenshots from this, and then I'm going and I'm asking you things like, what is this cell? What is this red fiber? You know, what is this purple fiber, right? What is this tissue right here? That's exactly what I'm doing. So you can make up your own practical just like I'm doing. And that's exactly how it's working. I'll have a little label. So what is the labeled cell? And so just to get, if you haven't done this kind of thing, looking at histology before, what you have to realize is that all these cells, you can't see the whole cell. All you can see is the nuclei, right? So the whole cells are something like this, right? But then you can see the nuclei here because you can't see the cell body, especially in connective tissue. You can see them sometimes in epithelial tissue, right? But the cells, there's a whole bunch of different sides. These are probably mast cells and stuff, but the only thing I'm going to ask you, your only option, if you recognize it as connective tissue proper, your only option for the answer of what is this cell is a fibroblast. So put that on your list of answers to what's that cell. All right. If I ask you what the fiber is, you have 2 choices. If it's red, almost always, it's collagen. If it's darker stained, this is the only sample where you could see it without being specifically stained for it. It's going to be elastin, elastic fibers or elastin fibers. Okay. These are long stringy things, right? So you look at this picture, it's a big mess, right? But you're going to be able to pick out collagen because it's going to be in every single organ we look at. There's going to be a layer of connective tissue in some sort. And this is the kind of junk you're going to see on these. So make a little chart, you know, a little PowerPoint for yourself, a bunch of slides, something like this, and label a bunch of stuff. And then make a list. What are the cells? Right, make a whole list of different types of cells you got to know as you move along right there, different types of fibers, different types of tissues. Right? So that is this one sample. A lot of the other samples for connective tissue, we're jumping right to the skin. We may as well, because we're going to go over, you know, a lot of the same ones are going to be seen in skin. So we'll go back to the slide box here, and you're going to go down to chapter 11, right? These are organized by organ systems. So you can go to chapter 11. And I've told your Bio 145 teachers to use this site, so hopefully they will. And then you'll be ahead of the game. So we're going to go to chapter 11, skin, and go to this 277, 278 right here, this thin skin sample. So while we're here, you know, I think I use this very sample, maybe not, but something that looks like it for your stratified squamous epithelial. And it's also in your skin exercise, you know, this is your thin skin, right? So this, all these cells, which are relatively close together, even if you can't see the whole cell, they're all kind of bound together. And because it's a dry stratified squamous, you got this dead cell layer on top there that due to preparation has kind of flaked off. So these cells are all stuck together. And then these cells over here are the fibroblasts, right, that are sitting in there. Are they really fibroblasts? Maybe not, but that's what you're going to call them, right? So epithelial and loose connective tissue, these are all, this is just stratified squamous dry. This is all a loose connective tissue, right? How do you know it's loose connective tissue versus dense connective tissue? Even though, because you wouldn't know it just by looking at it, right? How you'll know about it is by location. Here's the epithelial cells. Epithelial cells are always sitting on a layer of loose connective tissue. Right? This isn't the best picture of it, but you could see down here, there's kind of like little strands of it over there, even if it's closely packed in, right? So the reason I'm using this one is not to look at this picture though, it's to look at this number 2. You see there's 2 pages right here. You're going to go to number 2 right here, and you're going to click on this, and then this sample has been stained so that the elastic fibers come out. So I'm interested in this slice because it's bringing out these elastic fibers. Again, in this case, this stained a little dark, but this is the epithelial layer. And then you move into the loose connective tissue. The black circles are the nuclei of the fibroblast, but then these long stringy things over here are the elastic fibers, right, that are embedded within your dermis, right? And this is what gives skin your elasticity, right? So again, here's my demonstration, right? Here's your skin. Over here, the reason I could pull it up and it doesn't rip off is because the collagen fibers. The reason it bounces back into place, right, is these elastic fibers right over here. You can imagine these being stretched out, right? And they're going to kind of create a tension, and they're going to snap back when you let go. So this gives skin, right, over here, as well as all over here, the elasticity of it, your elastic fibers. So if I ask you what the black fibers are, you say elastic fibers. If I ask you what red fibers here, these big bundles right here, these are collagen fibers, right? And then this is dense irregular because this one's going this way. These are going the other way, right? They're running at kind of big bundles running cross angles to each other. And you get these different views of them, right? But this picture is good for when you're asked to identify elastic fibers and collagen fibers. What's the red stuff? Collagen. All right. And when you get to skin, right, the stuff right under here, that loose connective tissue is going to be called the papillary layer, which we'll talk about a little out. When you get to under here, this will be the reticular layer, right? These are two sublayers of the dermis. All right, so that's loose connective tissue, two different samples of it. This is the more like kind of stuff we'll be looking at throughout the entire semester. Then you got adipose tissue, which I'll use on this one. I don't know if this is the exact sample I use, but it'll be something like it. So this is all collagen fibers, loose. These are all this big bundles of dense, irregular connective tissue, the collagen fiber bundles. And then you come to these big, I should mention again, all these spaces that you see like this are actually artifacts. These spaces were actually adipose cells, right, that were filled with lipid before. And if you keep on moving deeper under your skin, you'll come to that layer that we looked in the cartoon, right, that yellow layer, or on the cadaver ones, right, that bubbly stuff, that yellow layer. This is the adipose tissue right here. So the big bubble is the actual cell, adipose cell. And because it's basically just for lipid storage, the nuclei is kind of pushed to the corner right there. You might see the nuclei over here. The rest of it would be in real life filled with lipids for storage. Right, so that's adipose tissue. And then there is, I kind of went over this dense, irregular, how to do it, and I'll show you one more slice of that. Let's go there while we're here. All right, so it takes a little getting used to, because again, it looks like, depending on what sample you're looking at, what was the other one? I believe this one I used. First, find the demarcation line between the epithelial layer over here, a bunch of cells stuck together. Right? And here you could actually see the cell membrane. Then you get to that loose connective tissue sitting right under it. And then as you go deeper, over here, you start to get the bundles of collagen fiber bundles, right? That's dense, irregular, going in different directions. You want to oppose that with, we'll go back to the connective tissue and to look at this dense, regular connective tissue. And in here, this is all collagen bundles. When we start to look at muscle, muscle looks a lot like that, but muscle is not in this unit. So I will not ask you about muscle tissue. So your answer should never be muscle. If it looks like this, it's dense, regular connective tissue. And can you see the contrast between that and the irregular? All these are bundles. They're all running in parallel with each other. These little lines, again, these are artifacts, right? The little purple things in here are the nuclei of fibroblasts. Anytime you see these purple things. Right, you notice, these may have a little bit of capillaries here and there, but there's, in contrast to the dense, irregular, or regular, there's no blood vessels going through there. This is why it's, white, basically. Basically, if you look at the white of your eye, that's going to be all connective tissue like that, kind of a fibrous tissue. And then that's the color of it because of the lack of blood vessels. All right, so that is your dense regular. You see how they're lined up with each other. So the other thing is that, these slides do, have little links to them, right? So you can kind of zoom in on some of them over there, right? Like, especially with the epithelial stuff. Okay, so, just to go back to that lab, because I didn't, I do have a video on it, but for the epithelial stuff, right? First off, there's this nice cartoon thing that kind of, in the cartoon version is very easy to identify these epithelial tissue, right? But in the real life histology, all you might see is the nuclei of these, and you're going to have to really determine, all right, these cells are pretty close together. They have a flat shape, so they must be squamous. They have a round shape, so they must be cuboidal. They have a columnar, like oblong, elliptical shape, so they must be columnar, or they look kind of really weird and they must be pseudostratified. So very simple to do when we're talking about the cartoon picture, but it's just not ideal. It's never perfect because of all these artifacts. Right, so for instance, here is, let's see, this is your lab activity. This is the epithelial one, where I give you a couple samples of different types of epithelium. And we're going to go down to this sample. Right. This looks a lot like, in fact, this is the same kind of sample. This is from that mesentery thing. Again, you don't have to know it, but this is the coating of that loose connective tissue that you saw. But this here, the microscope is focused on the outer epithelial layer. So you can see these are the individual epithelial cells. They're all kind of connected to each other. Right, in this case, we're looking down on a sample right over here. It's like just a scraped off sample that we're looking down on. It hasn't been cut. Right? And so you're looking at the top layer right here. And this is a simple squamous, as it turns out. And you can kind of see underneath all these other nuclei are from the loose connective tissue under it. But the reason you know it's epithelial are you can actually see the cell membrane boundaries all connected to each other like this. And this is very characteristics of a simple squamous, this kind of egg, like fried eggshell shape over here. So that's your pavement epithelium. There's a couple different samples of your simple squamous epithelia. Simple meaning one layer, squamous meaning a flattened cell. Right, and so in this case, you could just click right to the simple squamous epithelia. Let me go back here for 1 second. Notice that all those samples that we're going to look at here are under the title simple squamous epithelia. So this MH16 has a bunch of simple squamous epithelia, but it's the same exact slide as this simple cuboidal. It's just that this one is going to link to simple cuboidal cells, whereas this one is going to have links to simple epithelia. So this eye sample over here, you have this link where you can zoom right in and find your simple squamous epithelia. Not that I'm going to use this very slice. I might use this one, right? It doesn't matter. I might flip the image a little bit so it's at a different angle, but once you've looked at this a couple of times and you have pictures of it for study, it won't be difficult. So in this picture, here is the epithelial lining. Here, this is the connective tissue underneath it. Here's the scattered cells and collagen. Right? But this is the back of your eyeball. These are the nuclei of cells. They'd all look something like this, like if we were able to see the cell membranes. Right?
So unlike for the connective tissue where we have to label for the practical, say the tissue, the cell, and the fiber, for
Right.
this one, it's just naming the type of epithelial tissue it is, right?
That's that. Yeah, there's no, the cells are just,
right.
In this part, yeah, there's no structure. Like when we get to bone next unit, there'll be like also structures and stuff you got to know. But for epithelial stuff, it's all just name the tissue straight.
Okay.
These are the answers right here, exactly as they appear in the lab manual is the easiest way to answer any of those questions.
Okay.
Like are taking your quiz and stuff, right? Yeah, so that's a good point, right? These might have different cells, they might have fibers, and they might have tissues, whereas these are just going to be the tissue. And then you just want to go to each slide that I'm giving right there. But these are the simple squamous, single layer, and the cell nuclei is not, it's like this kind of, right? A little flattened because the cell's like this. It's not like this, where the cell would be like this. And it's not like this, where the cell would be like this, right? These are simple cuboidal and simple columnar. Right? You want to look for this flattened cell. And you know that, I guess some people said that the cells wouldn't be arranged like this, lined up like this. They're going to be aligned. I guess that's not intuitive. So that could be confusing, but these are a simple squamous cell. Just get used to looking at this slide right there. So, this one is linking to this simple squamous cell, but this is the exact same one you're going to use for stratified squamous. So when we go through the eyeball tissue here, the cornea of your eye, the whites of your eye, On the outside surface, facing the environment, is a stratified squamous because you need a little bit of protection right here. Right? And this is really the only mucus membrane here that's actually facing on the outside environment. So you got tears and stuff running down here. Right? But for your stratified squamous, which isn't linked here, but if you go to the stratified squamous heading, you'll find the same sample, but you could just click on stratified squamous. So here it is, right? Here's that stratified squamous layer, right, from here to here. Right? And it's called stratified squamous, even though these cells look round like a cuboidal cell. Right? In fact, they are almost very much shaped like a cuboidal cell. But the convention for naming stratified tissue with more than one layer, is to look at the apical surface and look at what shape they are. Those are squamous, flat. Squamous, you could think of squat or squashed, right? And that's how we kind of categorize this tissue as stratified squamous epithelial. And it's a non-keratinized or wet because these cells are alive right here. These nuclei are darkly stained and they're alive. And so we'll contrast that when we get to the skin stuff. We'll see that all the cells in the very top layer are dead. All right, well, everybody with me so far? I'm jumping around a little bit from sample to sample, but because this is all about this one. The only other one, those are pavement epithelial eye, and one of them that's not going to be labeled in this chapter 2 one, because I want to point out, because they're really nice samples of blood vessels. So simple squamous cells are going to be lining your blood vessels. And this is a layer over here. It happens to be in connective tissue. And it is in this fetal pig snout. Remember, if I ask you what the tissue is, I'm not asking you if it's a fetal pig snout. I'm just asking you epithelial, connective tissue, whatever, right? So in this sample though, which does have a stratified squamous, because that'll be part of the skin, it's got bone, it's got cartilage, but what I'm interested in are all these blood vessels right here, because you could really kind of clearly make these out. They stand out a little better versus normal connective tissue. So you see blood vessels, the cells lining the blood vessels, and this could be anywhere. You could look at all these various blood vessels around. The cells lining it are going to be those simple squamous cells. And so here's the nuclei of it. Let's make a different color, blue. Here's the nuclei. And of course, not all of them are going to be in plane with this particular sample, but there's a couple, maybe there's another one, and then their cell body, is going to be, they're all going to be connecting somehow, somewhere. If you don't believe me, just look at the cartoon picture. Right, you won't really see them. They're going to be these cells that are lining, right? So the blood's going through here, and then stuff is diffusing out through the tissue around it, right? So any of these samples, all you got to be able to do is recognize these samples that I'm giving you right here and know, oh, he's looking for simple squamous. I'm looking for the blood vessels. If we could see that, what's the tissue type? Simple squamous epithelia. A lot of people are saying, let's make a group chat. I highly encourage that. All my other classes, those people do much better. Share stuff. You could do quizzes together, right? Talk over them, stuff like that. And then I did, this is incorrect. Cross this off. There's no electron. E34 refers to something else. So, the other types are going to follow the same pattern, right? You're going to go mostly back to, for the most part, they're all going to be under either the skin, well, in the case of connective tissue, or in this epithelial box, chapter 2. And in that case, you want to go to the simple cuboidal heading and go to this MH216, as opposed to this MH216, which will link you to simple columnar epithelial. So you go to the simple cuboidal one. And I point out in here, the ones lining the kidney tubules. So if you go to this one over here in the kidney, you go to the renal corpuscle over here, and this is one structure that has simple squamous, but all these tubules, which is the specialty of simple cuboidal, are made up of simple cuboidal, right? So all you could see really, it looks like a bunch of circles in a totally one kind of tissue, but actually each one of these, let's see if I could find a better one. You can actually see the, not really. One of these days I'll find a better sample. Well, all these nuclei, all these tubule type things in the kidneys are made up of simple cuboidal cells. And if you could see them, if you look at the cartoon pictures, right, they're going to look, let's make this blue, right? They're going to be like this, all connected to each other, but the nuclei in the middle, right, is going to give it away as simple cuboidal cells, right? All right, everybody kind of with me? Any questions so far on that? And you'll just follow that suit. I don't want to go all night here again. I know people get burned out, but that'll be the general program of how you want to go about these lab activities. And just try to find the slide that you're looking for, go over them, make your own tables. And some points I did start writing the tables in here. I got to fix just in the connective tissue one I didn't do. All right, so just go over all these samples right there. And on Thursday, we'll go over a little bit more of the skin stuff right there. Another good thing to do if you don't want to do it, get all this stuff from the chat, is just go on the discussion board in Canvas, right? And put your information up there, like say, who wants to form a study group? Here, I'll put the discussion right now, I'll start it. But you could just add a discussion and say, study groups, who wants to start? So now anybody can make up these discussion groups, anything you want to do. And you could reply here if you're missing any information, right? Whatever people are using this Discord thing, you young kids are going to find some way to pass information on to each other. All right, so I am going to stop there.